Thanks everyone for joining us at today's medical interpreter training session in the language of genetics and genomics. Before we begin, I'd like to acknowledge the traditional owners of the land on which we meet today, wherever we are. And on behalf of Queensland Genomics, I pay our respects to elders past, present, and emerging. And in doing so, I recognise the valuable contribution that First Nations people make to our society. My name's Katrina Cutler and I'm the Manager of Communications and Engagement at Queensland Genomics. A part of that role is to look after the Queensland Genomics Community Advisory Group, and this project was identified and led by the Community Advisory Group in a very close partnership with QIMR Berghofer and Genetic Health Queensland. I'd just like to introduce you to the project team. You've met me. Dr. Lindsay Fowles is a genetic counsellor with our statewide public genetic health service, Genetic Health Queensland. Lindsay is also on the Queensland Genomics Community Advisory Group and she's one of our presenters today. Dr. Miranda Vidgen is a medical genomics researcher at QIMR Berghofer. And Miranda has worked with the Queensland Genomics Community Advisory Group for a number of years now, providing us with scientific know-how and assisting us in delivering community-led projects. Priya Romero-Milne works alongside Miranda at QIMR Berghofer, and she's extending her scientist skills today to be our Zoom master. And Kate Sullivan works here with me at Queensland Genomics. She's our Senior Communications and Engagement Officer, and she's been looking after the communications around this event. So I'd just like to acknowledge the work of the team to bring today's event together. So what is Queensland Genomics? We're funded by Queensland Health to accelerate the implementation of genomics into Queensland's healthcare system. The integration of genomics into everyday healthcare has the capacity to transform health systems and health services all over the world with faster diagnosis, new treatment, and more cost-effective service delivery. Queensland Genomics is a $25 million Queensland government program running from 2016 to 2021. And it brings together genomics researchers, clinicians, healthcare administrators and patients to fast-track the implementation of genomics into everyday healthcare for Queensland. We invest in projects to build infrastructure and capability to support genomics in areas such as clinical workforce education, establishing genomic testing capability, designing systems for genomic data management, investigating the legal and social implications of genomics in healthcare, exploring the value of genomic medicine, not just in dollars, but in patient outcomes, creating community awareness about genomics in healthcare, and piloting the use of genomics with patients in different disease areas, including rare childhood diseases, epilepsy, immunology, cardiac care, sepsis, superbugs, and a range of cancers. We're working with Queensland Health, hospital and health services, researchers and the community to make sure that any change is implemented into the health system safely, effectively and equitably. Now today's session is mainly focused on the work of Genetic Health Queensland, which is Queensland's public statewide specialist genetic health service. But our presenters will also take us through some concepts used in other specialty areas, as we see genomics increasingly used in other health specialties. The agenda for today's session, soon I'll hand over to Dr. Miranda Vidgen from QIMR Berghofer to talk us through Genomics 101, an introduction to the science and some genetics and genomics terms. Then Dr. Lindsay Fowles from Metro North HHS or Genetic Health Queensland will talk us through genetics in the clinic and introduce us to inheritance patterns and results from clinical testing. So without further ado, I'll hand over to Miranda for her presentation. Thank you, Miranda.
So when we do this session face-to-face, what we usually do is icebreakers in order so that everyone in the room gets to know one another. This is a little bit difficult online. So I'm going to do two things today. Firstly, I'm actually going to start by showing you a participant language map. And so this represents languages interpreted by participants attending the 3 sessions that we're presenting. And my apologies if your country is not highlighted here. As you can see, there are a lot of countries represented on this map. Now, the top participant languages for people coming along to this are Mandarin, Vietnamese, and equal third is Arabic and Spanish. And that represents about 50% of participants. but we actually have 66 different languages and dialects interpreted by participants, including spoken and sign languages. Now, 29 of these languages and dialects have only one participant attending across the 3 sessions. It's because of this diversity in the languages that we're presenting this in English. So the aim here is to introduce you to English words that are used in genetic discussions, and then that gives you an opportunity to then take that information and consider it in the context of the language that you interpret. Most people are either new to the topic or only have a basic knowledge, and this is perfect because we're going to go through very basic concepts in genetics and genomics. In fact, for those people who maybe have a little bit more knowledge of genetics and genomics, you actually might find that we're skipping over some of the more unusual details. So we're giving the basic overview, but if there are exceptions to the rule, which often happen in biology, we may not explore those in detail. So if you don't have any existing knowledge, that's perfect. So the presentation I'm going to give you now is Genomic Terms, an Introduction to Concepts in Genetics and Genomics. The main concepts that I'm going to be doing in this presentation are basic biology around cells, genomes, chromosomes, DNA, and genes. But right at the end, I am going to look at a couple of different health problems that are caused by DNA, so you get an idea of what we're going to be talking about in Lindsay's section. So, to start with, an adult human has trillions of cells in their body. And cells are the building blocks that make up our tissues. So that can be skin or bone or blood. There are all different types of tissues. Cells function differently depending on their role in the body. So we have lots of different cell types, we have lots of different tissues. Yet despite these differences in function, cells have one very important thing in common. Inside our cells is the chemical DNA. Now, our DNA tells cells how to function, and there is the same copy of DNA in every cell in the body. So when we talk about a person's complete set of DNA, this gets referred to as the genome. So that's where this term comes from, genome. It's the complete set of DNA that is in a person's cell. So my DNA is unique to me, just as your DNA is unique to you. And DNA is actually part of the reason why we look a bit different. And it's why our bodies can function a bit differently. So if we look at this diagram on the slide, we have this blue circle, which has a diagrammatic representation of DNA. Now, DNA is a chemical structure with a backbone, which is this silver part or grey part that runs down the back of the coloured parts. And this just holds everything together. But it's the chemicals in the middle, these coloured little bits. Now, depending on the orders of the chemical, this communicates how cells function. Think of it like the order of the chemicals are like words in a book. Depending on the order of the words dictates what information is being communicated. Now, structurally, DNA is very, very thin. You can't see it with the human eye. You need microscopes and things to be able to see it. But even though it's very thin, it's very, very, very long. Imagine it like a piece of string or wool. It's for this reason that DNA coils tightly to form chromosomes. And the formation of chromosomes is this really special process to organise DNA so that it fits inside cells and doesn't become a tangled mess because it's really long. In humans, our DNA forms 23 pairs of chromosomes, and that's going to be something we come back to a lot. If we look at the white box down the bottom of this particular slide, we'll see that there are 22 pairs of autosomes. Now, the word autosomes just means they're not sex chromosomes. Everybody's got them. But we also have one pair of sex chromosomes. So in this particular image on the slide here, we have an X and a Y down in the corner here, which indicates that this is a male set of chromosomes. If this was X and X, it would be a female set of chromosomes. And as the name suggests, our sex chromosomes determine if we are biologically male or female. So in humans, almost all DNA is packaged up into 23 pairs of chromosomes. Now, I keep saying that pairs of chromosomes. This is because we have 2 copies of our DNA in each cell, and they each form this set of chromosomes. So if we see here, chromosome 1, we have 1 copy and we have a second copy. So, and that 2 copies becomes really important, especially in Lindsay's presentation later on. So it's something worthwhile keeping in mind. Now, I've said all cells contain the same DNA. Then how do they function differently? How do they become different if it's all the same DNA? This is because of genes. Genes control how our cells function. You will remember that I said that the order of the coloured parts in the middle of the DNA diagram communicate how cells function. A gene is a unit of DNA that controls the development of one or more traits. Now, when I say traits, that can be anything really. So if you think about your hair, is it curly or straight? Unless you happen to go to the hairdressers. That's a trait. Your blood type. So if you're AB or O, that's a trait. In the human genome, we have between 20,000 and 25,000 genes. Now, the reason for the variation in number is because we actually don't know everything there is about human genes. And it's important to note that, you'll hear this time and time again, we have 2 copies of each gene. And that is related to the fact that we have 2 chromosomes, because we have 1 copy on one chromosome and another copy on another one. So we've looked at a few new words, but what I think is really important to recognise is these are not common terms in the English language. Therefore, clinical geneticists and genetic counsellors often use analogies with English-speaking patients to communicate these concepts. So this is something that they do all the time. So I'm actually going to present a common analogy, which is basically going back over the information that I've already presented here today, just to give you an example of what might happen in a session with a genetic counsellor. And the analogy I'm going to use is the library analogy. And this kind of makes sense because libraries contain lots of information, as do genomes. So in this analogy, the library represents the genome, the source of all information. Books within the library are organised onto bookshelves, just like DNA is organised into chromosomes. So our books represent our genes, so that unit of information. And the pages of are our DNA, with words being that information communicated, those coloured little bars that we saw on the diagram. So if I wanted to learn how to cook, I wouldn't read all the books in the library. I would go to the bookshelves with the cookbooks and take out a few books. I would read through those books and learn how to cook. It's the same with our cells. We don't use our whole genome for every cell. If my cell needs to be a skin cell, it goes to the chromosome with skin cell genes. It takes those genes, it reads the information, and it uses that to become a skin cell. So that covers off on the basic terms we're going to be using. But I want to, in this next section of the presentation, we're going to have a look at how DNA causes health problems. But before we can discuss this, we first actually have to talk about where our genes come from. Our genes come from our parents and are passed on to our children. This is why some traits are similar within families. Earlier I said almost all cells have 2 copies of DNA. Now, a really important exception to that is eggs in women and sperm in men. In these cells, there is only 1 copy of DNA. So that when they come together, the sperm and the egg, to make the child, the embryo, this child has half the DNA from mum and half the DNA from dad. And since your DNA comes from your parents, variations that are in that DNA are inherited. Now, I said the word variations. Variations are changes to that chemicals in the DNA, so that order of the chemicals. So those coloured parts that I showed you earlier. Some variations are normal. They're just part of being a human being. They're neither bad nor they're good. They just are. But there are some variations that cause problems. Think of them like spelling mistakes and spelling. Sometimes there is a mistake that means you can't understand what is being said. And there are other times where you can still read even though when there's a mistake present. So not all variations are bad or good. Some create changes, some don't create much of a change. Some are good, some are bad. But this brings us to the health problems that are caused by our DNA. Now, our first one here is inherited variations, which are conditions that are a result of DNA that comes from the parents. Now, this will actually be the focus of Lindsay's talk later, so I'm not going to go into it in any detail. But what we need to recognise, and what's important to recognise, is that not all DNA-based health problems are the result of inherited conditions. We also acquire variations in our DNA over our lifetime, which are these ones here. So this can be from exposure to a lot of different things, from chemicals to radiation. Or it can simply happen due to errors when our cells are duplicating themselves. So you'll probably come across variations tend to cause diseases like cancer. There are also conditions which are multifactorial conditions. Now, these conditions are when we have an inherited predisposition, but having that inheritance alone doesn't actually cause the disease. We also need things like lifestyle factor and environmental factors for the disease to develop. Finally, there are chromosomal disorders. When our cells duplicate, which they do all the time as cells die off and also as we grow, DNA is copied. So we do a complete copy, and then one copy goes into one cell and the other copy goes into the other cell. And sometimes a piece of the chromosome or part of the chromosome is missing or duplicated. Now, if this occurs when sperm or egg are being formed, so when we split our DNA in half and half, or when a baby is in that very early stages of development, when it's a tiny, tiny little embryo, and they don't get a complete set of DNA, either too much or too little, this sometimes, depending on what proportion of the DNA goes into the cell, can cause health problems. So that's what we're going to talk about on health problems for the moment. And I think we can get started with Lindsay's presentation if you're ready, Lindsay.
I'm going to talk more on the specifics of what we talk about in a clinic, and that inheritance. So when we first meet with one of our patients who's been referred to genetics, there's a couple of things we need to find out. We need to find out about their personal medical history, and we also want to find out about their family history. Now, not everybody has complete information about their family tree, and that's fine. But if they do have information, we'll ask about it and record it. And I'm going to talk about how we record family trees as well. Then I'm going to move on to genetic conditions. And when a genetic condition it can be inherited through the family, or in some cases, it's happened in an individual for the first time. So sometimes it's inherited and sometimes it's happened for the first time, and that's what we call sporadic, just out of the blue. When it is inherited, there are a number of different inheritance patterns that can occur, and this is why we draw the family tree, because the pattern that we see in the family tree can give us some clues about what type of inheritance it is. And that can give us some clues as to what the underlying genetic cause might be. And I'm just going to review 3 of the most common inheritance patterns we see. This is not a complete list of inheritance patterns, but they are the three most common. Then I'm going to come back again and talk about when people do have a genetic test, what are the possible outcomes? We find an answer, we don't find an answer, we find something we don't understand. And then when we do find an answer for their condition, that's called a pathogenic mutation. If we do find the mutation that is the cause of their disease, a pathogenic mutation, what does this mean for that person? What does it mean for their family? So those are the 5 topics I'll be covering. So as I said, when we first meet a patient, the things we ask them about are their medical history. And we want to know what it is that's brought them to the genetics clinic. You know, why did their doctor think a referral to genetics was a good idea? What is it in their medical history? And we'll ask about the condition, the medical condition that they have, how it's affected them, how severe it's been, and how old they were when those signs of that condition appeared. And then we'll ask them about their family, you know, their mother, their father, their brothers and sisters, any children they may have. And we'll actually go all the way back to their grandparents and their aunts and uncles. So yes, we like to record family trees. And as I said before, when we record the family tree, it also helps us identify the patterns that might tell us what type of inheritance pattern we're seeing in the family. So this is how we draw a family tree. And you can see here, this is what we call, this would be a basic family. There's a father, so we use squares to represent men. A mother, we use circles to represent women. When there is a line connecting them like this, that goes from the middle of the square to the circle, that means there's a relationship. So whether they're married or in a relationship together. We try to keep the same generation on the same line. So this is the parent, and here are their children. And this type of connection indicates that they are children. So that this husband and wife have one daughter and one son. Sometimes we'll have a very big family, or people won't have complete information about a family. And we'll use a diamond shape to say, we know that there is a brother or a sister, but we don't know whether they're a boy or a girl. Or perhaps we know that mum has 8 brothers and sisters. We just want to write down that there are 8 siblings. And we'll write down a triangle and write a number 8 in the middle. And so that just tells us it's a big family and there's no health issues of concern in those people. If we've drawn a line through an individual like this, it means they've passed away. Now, to record what particular medical conditions are in the family, we'll use symbols like this. And I've just used the 4 basic ones, but you can use all sorts of different things. You can put circles, you can put spots, you can draw a flower in there. It really doesn't matter how you do it, so long as you write on it somewhere what that symbol means. And this is just an example. In a family, I might use drawing in this lower right corner as meaning it's a person with intellectual impairment, or the bottom left corner to mean they've got hearing loss, they're deaf. But this can be different for any pedigree, for any family tree you draw. You just have to define it for each family tree. So when we've drawn that family tree and we've seen that there's a condition running through the family perhaps, this helps us understand what the inheritance pattern might be. And I've said I'm going to cover the 3 most common. We've got autosomal dominant, autosomal recessive, and X-linked recessive. So autosomal dominant is the most common one. Now, this autosome just means it's a chromosome, but it's not one of the sex chromosomes. So we have 23 pairs of chromosomes. 22 pairs are autosomes. They're the same for boys and girls. It's just that last pair, the sex chromosomes, that are different between boys and girls. Girls have 2 X and boys have an X and a Y. But autosome just means it's somewhere on those chromosomes that are the same between men and women. So there's no difference in inheritance between men and women. Dominant means that the mistake that's being inherited through the family. Now, if we have 2 copies of the gene, well, 2 of the chromosome, and this chromosome carries a broken copy and this chromosome carries a good copy. If you're affected by the condition, you've got 1 broken copy, 1 good copy, that's autosomal dominant because the broken copy is dominating over the good copy. And so you know the instant someone's got a broken copy of that gene because they have the condition. Their broken copy is being the most dominant copy. We thought in autosomal dominant, we can see all the people here who carry the broken copy of the gene because they have the condition and it's being passed down from the grandparent, and he's got an affected daughter and an affected son, and they're passing it on to their children as well. And you can see there's a mix of men and women. And this is here's a pair of chromosomes where we've got the genes marked on it. Now, all the way along the length of this chromosome are lots and lots and lots of genes. There's hundreds of genes, possibly thousands, depending on which chromosome we're looking at. And the chromosomes come in pairs, so we all have pairs of all of our genes. Now, I'm only going to focus on 1 gene on this chromosome because it's the gene that's involved in this autosomal dominant condition. And on this chromosome, the red bar is the broken copy of the gene, and this is the copy of the gene that's normal. It's the usual copy. This person is affected by the condition because their broken copy gives them the condition. The good one isn't enough on its own to keep them well and healthy. So here we have this is how it is inherited. This parent is affected by the condition. Half of their chromosomes carry a broken copy of the gene and half carry a good copy. Now, the other parent is unaffected, so they only carry 2 good copies. Now, when our children inherit their genes from us, we don't want to pass on both 2 copies to our children, from the mum and from the dad, because then the baby would get 4 copies, and that's bad. So the body has a really neat trick. So when the mothers make their eggs or the men make their sperm, the cells have a really neat trick where all the chromosomes get lined up in pairs and then it gets split down the middle. And just 1 of each pair goes into the sperm and just 1 of each pair goes into the egg. So the sperm or the eggs only carried 1 copy. So if this were the father, then half of his sperm would carry a broken copy of the gene and half of his sperm would carry a good copy. Same thing that if this were the mother, half of her eggs would carry a broken copy of the gene, half of her eggs would carry a good copy. Now, there's 4 possible ways in which the egg or sperm from this parent can combine with the egg or sperm from this parent. If this egg or sperm is involved, then we're going to get a child who's inherited a broken copy, and they will be affected by the condition. So you can see it doesn't matter which one of these was involved in making that child. They get 1 broken copy from one parent and a good copy from the other parent. And these 2 possible combinations will result in an affected child. If, however, this egg or sperm is involved in making that baby, then, and it combines with either one of these 2 options mean the child will be unaffected, doesn't have the condition because they've got 2 normal copies of the gene. So of those 4 possible combinations, there are 2 which give you an affected child, 2 combinations which result in an unaffected child. So for every pregnancy, every time an egg and a sperm come together, we have a 1 in 2 chance of an affected child. And I'm just going to come back to that this is the same slide I showed you before, exactly the same. So now you can see how it's coming from a parent to a child and then down to the grandchild, or the grandparent to their parent to the child. You know, it's just being passed down through the generations. So someone asked the question about why does the bad dominate, why not a good one? And it really depends on the condition. In some conditions, the bad dominates, and in some cases, the good dominates, and that's what autosomal recessive is. So that was a really excellent question. This is autosomal recessive is when the good dominates. And what actually happens here is that the broken copy is hidden, and we call it recessive because it recedes behind, or it hides behind the good copy. So people in this generation and in the grandparent generation all carry the gene, all carry a broken copy of the gene, but they've only got one broken copy of the gene. And we can't tell that it's there because their good copy keeps them well and healthy and hides the bad copy. That would be awesome if we could find out which egg or sperm carry the broken gene, but that's kind of tricky. And there are options in IVF to try and find the embryo which carries the broken copy, but no, we can't pick out which egg or sperm do. In autosomal recessive, we will only see affected people when there's a broken copy of a gene and a broken copy of exactly the same gene in both sides of the family, so in both the father's side and the mother's side. And if the mum and the dad both carry one broken copy of the gene, so one broken copy, one good copy, there is a chance, not definite, but there's a chance that they will have a child who inherits a broken copy from mum and a broken copy from dad, and they'll be affected by the condition. This word here, consanguinity, just means that the parents are related, so that they come from the same family. And in some cultures, this is very common. Perhaps cousins marry, and because they are from the same family, it means there is a slightly higher chance, again, it's certainly not definite, but it is a slightly higher chance that they will carry the same broken gene in the mum and the dad. So this is autosomal recessive inheritance. We've all got 2 copies of the gene that's involved, but for a person to be affected, both of their copies have to be broken. For a person who carries a normal copy and in the other copy, their other copy is broken, they are well and healthy, they don't have the condition, but that we call them a carrier because they carry a broken copy. They're not affected by the condition, but they carry one broken copy. And they probably, and in most cases, people don't know that they're a carrier for the condition. And in fact, everybody, all of us here today are carriers for different conditions. We all are carriers for about 6 or 7 different conditions. And you'll only have an affected child in the small chance that your partner is a carrier for exactly the same condition. So when 2 carrier parents meet, and generally they'll have no idea that they're a carrier for this condition. Each parent, half of their eggs for the mum will carry a good copy and half of their eggs will carry a broken copy. And for the dad, half of their sperm will carry a good copy and half of their sperm will carry a broken copy. And then there's 4 possible ways in which those eggs and sperm might come together. So if we get a sperm which has a good copy with an egg which has a good copy, then that child is completely unaffected and also not a carrier. So only carries 2 normal copies of that gene. If we've got a good copy from one parent and a good copy from the other parent, 100% of the population are carriers, but we're all carriers for different conditions. So carrier for a specific condition is usually very low. Or if they've inherited the broken copy from this parent and the broken copy from this parent, then these two possible combinations are also well and healthy, but they're unaffected. It's only in the chance, the 1 in 4 chance, that we get this particular egg or sperm combining with this particular egg or sperm that a child inherits 2 broken copies of the gene and they have the condition. So you can have 2 carriers for a condition marry and have children and have completely unaffected kids because they just haven't had the situation arise where this has happened. And sometimes we'll have 2 carriers who meet and have children, and they'll have quite a number of their children affected. It's completely random. It's an independent chance for every pregnancy as to what combination will come together. So we'll come back to this, exactly the same slide I showed you as before. Here's a family with an autosomal recessive condition. There's a broken copy of the gene in these people as well, but they've only got 1 broken copy, so they're all well and healthy. It's only now that we've got a father and a mother who each have a broken copy of the same gene. Remember, we've got 25,000 genes, so it's pretty unlucky, but it's possible to meet someone who's got a broken copy of exactly the same gene, and they've now had affected children. They can have boys or girls. There's no mix in it. It doesn't matter whether you're a boy or a girl. It's an equal chance. This one gets a bit trickier because this is when sex becomes a factor, and we talk about X-linked inheritance. Now, before we were talking about the autosomes, which are the same for men and women, but the sex chromosomes, women have 2 copies of the X chromosome, whereas men just have 1 X chromosome and a Y chromosome. Because they've only got 1 X chromosome, they've only got 1 copy of the genes that are present on, that are exclusively on the X chromosome. So if they inherit a broken copy of that gene on the X chromosome, they're going to be affected. But women are a bit luckier in these conditions because they've got 2 copies, which means they've got a second copy to help keep them well and healthy. The important thing to note in X-linked conditions is that a father never has an affected son, never. Their daughters may be carriers of the condition, but their daughters won't be affected. So a father will never have an affected child. And their boys, yeah, I'll go into the specifics again. So here we go. We've got a male here, and we're just looking at the pair of the sex chromosomes. This is an X chromosome, and the Y chromosome is smaller. And this is the gene we're interested in, up the top of the X chromosome here. If a boy has a broken copy of that gene, they haven't got a second copy, so they will be affected by the condition. But women, they've got 2 copies of this gene. So if a woman carries a broken copy of the gene, as you can see here in red, they've also got a good copy of the gene here in white, and that helps to keep them well and healthy. Now, this is a little bit of a simplification because women who are carriers for an X-linked condition may show mild signs of the condition, and if people out there may have more understanding of X-linked and may say, but sometimes women can have significant signs, and it's true. But as a general rule, the women are only very mildly affected or not affected at all. So that because they've got 1 copy helping keep them well and healthy. When we have a woman who is a carrier for a condition, but the father is unaffected, what are the chances that their child will be affected or be a carrier? So when the sex of the child is controlled by which sperm was used to make that child. Now you can see that with an unaffected father, the only thing we're interested in here is whether there's an X chromosome in the sperm, or a Y chromosome in the sperm. If there's an X chromosome in the sperm, it's one of these two possibilities. It will be a daughter because they've inherited an X from their dad, and they can only get an X from mum. And we've got that 50-50 chance. Did they get the egg which carried a good copy of the X chromosome from their mum? Or did they get a broken copy of the X chromosome from their mum? So it's that 50-50 or 1 in 2 chance that a daughter will be a carrier for the condition. And so she'll just be like mum and most likely unaffected. But when it comes to the boys will always inherit their Y chromosome from their dad. That's the only way they can end up being a boy. Because to be a boy, you have to get a Y chromosome, and the only place they can get it from is dad. So anyone who's inherited a Y chromosome will be a boy. And then we have that same question. Which X chromosome did they inherit from mum? If it was the good X chromosome, then that will be a boy who's not affected by the condition. Or if it's the X chromosome which carries a broken copy, they'll be a boy who's affected. So hopefully with that explanation you can see that boys or fathers never pass the condition on to their children, like they never have an affected child, but they can have a carrier daughter. So their daughter is at risk for having an affected son. And you can see here that all these affected boys are connected through their mother's line. So this boy goes up to a woman, up to his mother, to her mother, and presumably this woman was a carrier as well. So she has passed on her affected X chromosome to this boy and her affected chromosome to this woman. So X-linked recessive conditions will see affected boys connected through unaffected women. All right, so look, just trying to help you reinforce, just to go over some of the things I've just spoken about. This is just to try and illustrate real life situations that these are actually families I've seen. So our patient was John, and in John's family there's a kidney condition called polycystic kidney disease. And you can see that it's being inherited through the family, that he's got 2 brothers who have this condition, his dad had this condition, he's got 2 aunts who've had the condition, as well as his grandfather. John's come to see us because in this condition, people's kidneys can fail and they need to go either onto dialysis or have a transplant. And his brother here has already had a transplant, and this brother here is approaching the point where he needs a transplant. And John is considering being a donor for his brother, like giving his brother one of his kidneys, but he really wants to know, does he actually have the condition? And we're going to try, and in this family, we're going to try and use genetic testing to clarify exactly what's happening in this family. What type of inheritance is it? And it's actually part of the name of the condition. Is it autosomal recessive? Is it autosomal dominant? Is it X-linked recessive? Or is it not inherited? And we know it's autosomal dominant because it's coming through the generation. So the broken copy of the gene is dominating. If a person has a broken copy of the gene, they have the condition. And you can see it's come from a father to both daughters and a son. So there's men and women inheriting the condition. And then this father only had 3 boys, and 2 of his boys have the condition, and we're uncertain about the 3rd one. We're just use genetic testing to double check. Okay, so let's get rid of that one. And here we go. So this time I'm giving you a bit of a clue. I'm telling you it's autosomal recessive condition. And we don't really need to know anything about this fancy name, except that the gene that's involved is called MUTYH. Again, you don't need to know that, but that's the name of the gene. And if people carry 2 broken copies of this gene, they have this condition called MUTYH-associated polyposis. Fancy big long words to say that someone gets a lot of bowel polyps. And I'll hear some of you saying, what is a bowel polyp? So polyp is just where a cell grows more than it should. So it's a growth on the lining of the bowel. Why do we care about bowel polyps? Because sometimes, not always, but sometimes a bowel polyp goes on to become bowel cancer. So if someone's getting lots of bowel polyps, it's useful to know about it so they can have extra screening. So if you know what a colonoscopy is, that's when the bowel gets screened for polyps and they can be removed before they become cancer. Now, Anna, who was 76, just happened to have a colonoscopy, and her doctor was a bit surprised when he discovered 60 bowel polyps. That's much more than we would normally expect to see. We'd only expect to see just a few in someone like Anna. And we did some genetic testing, and we confirmed that both of Anna's copies of the MUTYH gene carry a mistake. So genetic testing has confirmed that Anna has this condition. Now, genetic testing hasn't been done in anybody else in the family, but we know from what Anna tells us about her family history that her brother Ben also has bowel polyps, so we don't know how many, and also Bob has been shown to have bowel polyps. When we asked her about her parents and their brothers and sisters, we're not aware of anyone having bowel polyps or bowel cancer. And again, she doesn't know much about her grandparents. That was a long time ago. So the question is, what are the chances that Anna's daughters are going to be affected by this condition? If we know it's autosomal recessive, which means you need to inherit 2 broken copies of the gene to be affected. The chance of Anna having a daughter affected by this condition is very low. It's close to 0%. It's very close to 0%, but it's not actually 0% because there's a small possibility that the father, either this father, now Anna's had children with has had 2 relationships where she's had children. This father has had 2 daughters, and this father has had a single daughter with Anna. And if either of these men were a carrier of a broken copy of the MUTYH gene, then there'd be a chance that these girls would be affected. But the chance of that is quite low. So it's nearly 0%. And we're going to go on to the third family history we're going to look at. Now this is something called Duchenne muscular dystrophy. And in this condition, boys are affected because it's an X-linked condition. And what happens in this condition is that they deteriorate with time. They're born well and healthy. It's often diagnosed around about the age of 4, 5, 6, 7, when the boys start moving a little bit differently. And it's noted that their muscles deteriorate with time. So while they're quite young, they're still able to move around. It's not uncommon by the time they're a teenager for them not to be able to move around and be in a wheelchair. And then they commonly pass away in their 20s because the muscle that controls their breathing stops working as well. It's a very difficult condition to have in your family. It's a devastating condition. And the person that we saw was Greta. Now, Greta's only 16, but she's looking ahead to the future, and she wants to know, she's trying to wrap her head around whether she wants to have children, what plans she might make for the future. She's not planning to have children at this time. She's just getting herself into a headspace where she's thinking about that. And we know that because she's connected through her mum to an affected boy, that there is a chance that she could be a carrier for this Duchenne muscular dystrophy. Now, we abbreviate that to DMD. So the boys who have the marking in the top right corner have DMD. Now, Carl was born quite some time ago, and he passed away before genetic testing was possible for this condition. So no genetic testing was done in this immediate family. But by talking with Greta and her mother, Lyndall, we find out that there is another branch of the family where there's been an affected boy born. I've just drawn attention to this up here because I haven't shown, this type of drawing indicates that Ilsa and Martha are identical twins. So just by chance, they identical twins have exactly the same genetics as each other. So we know that both Ilse and Martha are carriers for DMD because descended from each of them is a boy who's been affected by the condition. We're not sure if Lyndall is a carrier, and because we can't be sure if Lyndall is a carrier, if she is, there's a chance that she could have passed it on, the carrier status, to her daughter Greta. And we'll only be able to know for sure if we can do genetic testing and confirm if Greta does or does not carry the mutation that was responsible for Peter and Carl developing the condition. Now, because Peter has been born more recently, he's the same generation as Greta, we're suspicious that genetic testing may have been done in Peter, and we encourage Lyndall and Greta to contact this branch of the family. The two branches of the family have lost contact with each other. But Lyndall makes the effort. She contacts Andrea, and we find out that Peter has had genetic testing, so we know the exact genetic mutation in this family that is responsible for the boys developing DMD. And we're actually able to test Greta and confirm that she doesn't carry that mutation, that she has inherited a copy of the X chromosome that carries a good copy, not the copy that carried the broken version of the gene. So through genetic testing, we've been able to confirm for Greta that she has no chance of having a son affected by this condition. But we know that the family is bigger than this, that there are other branches of the family. And my question now is, who else in this family might benefit from genetic testing to confirm if they are or are not a carrier for this condition? Who else has a chance of having a child affected by DMD? Is it Peter? And the reality is with DMD that the boys won't be having children, but if Peter were able to have a child, could he have an affected child? Is it Lara over here? Is it Peter and Lara could have a chance? Or could it be Edith? Now, I'll just draw your attention to the fact that Edith is in her 70s, and Edith has had 2 daughters, and she's unlikely. What are the chances of someone in their 70s having another child? X-linked inheritance is tricky to follow. But the important thing to remember is that the broken gene is carried by women, and the women are generally unaffected. It is true that the female carriers with DMD can have some health impact, but they don't end up like the boys, where the boys end up in wheelchairs in their teens and passing away in their 20s. It's actually possible the fact that Edith in her 70s is in a wheelchair could be connected with it. But that's the type of thing we're talking about. You know, the boys pass away in their 20s, but a woman can be in her 70s. And that's very unusual to find a woman ending up in a wheelchair because she's a carrier. These 2 women are of a similar age. They are definitely carriers, and they're not showing any signs. But you can see that there's a link through the women. So we go to Edith, to her daughter, down to Lara. So yes, Lara is at risk. Lara only as being the one who's at risk for having children who are affected by DMD. Now, Peter's not at risk for having a child who's affected by DMD because only boys are affected, and he can only pass on. If he could possibly have children, then his boys would inherit his Y chromosome, and his Y chromosome doesn't carry the broken copy of the gene. If he passes on his X chromosome, then his daughters would be carrying. Edith was at risk for having an affected son, but she's now past the age of having children. So she's had 2 daughters. Both of her daughters are at risk of being carriers, and we know that Lara is at risk of being carriers. So I think we'll leave it at that for the moment. So when people have testing, we focus either on the specific genes, or sometimes we look at the complete chromosome. But when we're focusing in on genetic testing, there's 3 possible outcomes. And this applies mainly when we're looking at specific genes. And I talk about the black, the white, and the grey possible and grey zone results. You know, we can have black and white, which are very clear answers, and sometimes we get a grey zone. So the first possible option is we find nothing. We don't get an answer. We're in exactly the same position we were in before we started. And it might be that the condition is not genetic, that there's nothing there for the genetic test to identify. But it can also be a limitation in our knowledge. We're looking in the wrong gene, or we're using the wrong technology, a limitation in the laboratory techniques. Another possibility is we get an answer, and that can be awesome to the family and the individual. It means we know exactly what the disease is. So if we know exactly what the condition is, the doctors have more information about how best to treat that person. That person understands what they have and why they've got it. And it also means they know what are the chances that someone else in the family might get the condition. So when that word recurrence risk, what are the chances that their children will inherit the condition? What are the chances that, and when we can test others in the family, we can offer testing to their brothers, their sisters, cousins, anyone in the family who might be at risk for developing the condition or being a carrier for the condition, and tell them if they are or they aren't. And if they know that there's a chance that a child could be affected, then this opens options in pregnancy. And that can be either through testing in a pregnancy or through IVF. I'm not going to talk about that today, and I'm not saying this is something everybody does, but it opens the possibility. Then we have this challenge of a grey zone result, where we find a variant and we don't know exactly what it means. This is just a fancy phrase for saying we found something different, we found a variant. Uncertain significance means we don't know what it means. And we talk about, everybody's got 25,000 genes, we're all human, and we are all different. So we know that there are a huge range of differences in our genes, which are normal differences. They're just part of what makes us different from each other. Sometimes we find a difference in a gene and we don't know if that difference is one of the differences that falls into the normal bucket, or if it's a difference that falls into the bucket where it causes disease. And if we find one of these differences, even though we don't understand what it means, we will tell the family, we will tell that individual, because in time, in 2 or 3 years' time, or 4 or 5 years' time, we might know, because our knowledge grows all the time. And if they come back to us in a few years' time and ask us that question, we may be able to answer it. But we can't keep track of all these variants. So we give that information to the patient and give them the power that if it's something they want to know about, they can come back to us. So those are your 3 possible outcomes: the black, the white, and the grey. Yes, we found something. No, we found nothing. Or we found something we don't understand.
Thank you, Lindsay. So just to wrap up of our intended outcomes from today's session. Today, you have received an introduction to terms used in genetic counselling sessions. So that includes the basic biology of genetics, inheritance of genetic conditions, and outcomes from genetic testing. I'd like to thank you all for your participation in today's session. And if you have any questions at all after the session, please don't hesitate to get in touch with us at communications@qgha.org or visit our website. You can see the website details there. We really hope you enjoyed the session and that this introduction to genetic and genomic terms has helped to build your knowledge and your confidence in your interpreter work in this specialist area. Thank you for attending and have a great day.